Imagine you're 35 years old. You are in constant, debilitating abdominal pain.
Yeah, the kind of pain that just forces you to cancel plans or take time off work.
Right, the kind that slowly starts ruining your daily life. So you finally go to the doctor, desperate for a fix.
And you go through a battery of invasive tests.
Exactly. And instead of the clear, actionable diagnosis, you're handed a label. Idiopathic chronic pancreatitis.
Which is really just the incredibly frustrating medical way of saying we have absolutely no idea what's causing this.
Right. But here's where it gets worse. Along with that vague diagnosis comes a massive, unspoken social stigma.
Oh, the alcohol assumption.
Yeah. Because traditionally, chronic pancreatitis has been so heavily associated with heavy drinking, almost everyone, sometimes even your own medical team, assumes you must be a secret alcoholic.
Even if you aren't.
Exactly. You rarely even touch a glass of wine.
Yeah.
Yet you're stuck with this stigmatized, incredibly painful disease, and doctors can't really treat it.
It is the absolute definition of diagnostic muddy waters.
It really is. Today, we're looking at a medical breakthrough that proves those long-held assumptions completely wrong.
And it highlights a tragic reality of this specific disease. I mean, the pancreas is deeply hidden in the abdomen. It's an incredibly volatile organ.
So by the time someone is experiencing that level of severe pain, the tissue damage is already profound.
Right. For decades, gastroenterologists and researchers have been trying to solve this puzzle while essentially completely blindfolded.
Because you can't just take a piece of a living inflamed pancreas out to study it safely.
No, you really can't. The organ makes highly potent digestive enzymes. If you mess with it too much, it literally starts digesting itself.
Okay, let's unpack this. Our mission for this deep dive is to decode how scientists finally remove that blindfold.
It's such a cool study.
We are digging into a truly groundbreaking 2026 paper published in the journal Cell Stem Cell by Osorio Vasquez and colleagues.
They achieved something that genuinely sounds like science fiction.
They built a biobank of living human pancreas tissue to find personalized cures. They basically found a way to take the pancreas out of the dark and put it under the microscope safely.
Which is exactly why I believe that understanding the why at a cellular level changes absolutely everything in medicine.
Yeah.
When you can finally see the actual mechanisms of a disease working in real time, you start to realize just how wrong our foundational assumptions have been.
So this research doesn't just shed light on the pancreas.
No, it completely rewrites our understanding of how chronic inflammation operates at a cellular level.
So let's start with the logistics. If we can't safely study the pancreas while it's inside the patient because it's too dangerous, we need a proxy, a stand-in.
Right.
How exactly do scientists get one without harming the patient?
They create what the scientific community calls patient-derived organoids, or PDOs for short.
Okay, for anyone trying to visualize this, think of these organoids as biological avatars.
I like that term.
They are miniature, three-dimensional versions of the patient's own organs growing outside their body in a dish.
And they aren't just generic cells. They retain the patient's exact genetic code, their exact mutations, and their highly specific disease profile.
Right. And the how here is fascinating. Traditional cell cultures involve growing cells flat on a plastic petri dish.
But human organs aren't flat.
Exactly. So researchers take a tiny sample of tissue. This could be from a necessary surgery or even just from pancreatic juice collected during a routine endoscopic procedure.
They extract the stem cells from that sample, and then they drop those cells into a specialized 3D gel matrix.
It's like biological scaffolding.
Exactly like scaffolding. This 3D matrix mimics the physical environment of the human body.
So when you feed these cells the right chemical growth factors in this 3D space, they naturally self-organize.
They do. They grow and fold into these tiny, hollow, spherical structures that perfectly replicate the intricate ductal network of the actual human pancreas.
That is wild. And in this specific study, the researchers successfully established a living biobank of 37 of these chronic pancreatitis organoids.
Right, an N of 37 cohort, which, might not sound huge in the context of a massive drug trial.
But for a living 3D tissue biobank of a rare disease, that is incredibly dense, hard-won data.
Absolutely.
Hold on, I need to stop you right there. We started this whole deep dive talking about the massive social stigma linking pancreatitis to heavy drinking.
Right.
If they just pulled 37 samples to build these living avatars, wouldn't the vast majority of them be alcohol-related? How does that math work out?
That is the exact stigma we mentioned, and this biobank completely shatters it. Let's look at the actual demographics of these 37 avatars.
Okay, let's hear the numbers.
43% of them, that's 16 patients, were driven by a specific genetic mutation.
Wow.
35%, or 13 patients, were idiopathic, meaning unknown origin.
So where does alcohol fit in?
Only 8.1%, which is just 3 single patients out of the entire 37, had alcohol-related pancreatitis.
Wait, only 8%. The entire medical narrative is flipped on its head.
Completely. And the remaining few were rare mechanical cases, like a physical obstruction or groove pancreatitis, both at 2.7%.
So this biobank definitively proves that the disease we're looking at is overwhelmingly driven by internal genetics and unknown factors, not lifestyle choices like alcohol.
Precisely.
And when you say genetics, the paper gets very specific about the molecular machinery that's breaking down, right?
Yeah, they fully sequenced the DNA of these avatars, and they found some major culprits.
Like what?
They discovered that 19% of the cohort had a mutation in a gene called PRSS1.
Oh, PRSS1. Let's use a memory trick here for the listener.
Yeah.
The PRSS1 gene codes for an enzyme called trypsinogen.
Right?
Think of trypsinogen as a pair of extremely sharp molecular scissors that help digest the protein in your food.
Normally, the body keeps those scissors securely closed inside a safety sheath until they safely reach your stomach.
But the PRSS1 mutation makes those scissors prematurely pop open while they are still inside the delicate pancreas.
Right, so it literally starts shredding and digesting the organ from the inside out.
That's a perfect analogy.
They also found that 16% of the cohort had a mutation in SPNK1.
Okay, so if PRSS1 is the sharp scissors, SPNK1 is the safety cap.
Exactly. When SPNK1 is mutated, the safety cap is fundamentally broken, meaning even normal scissors can run wild and cause damage.
But the most prominent mutation they found, the one we really need to focus on, was in a gene called CFTR. Right.
Yeah, a massive 27% of the entire cohort had a CFTR mutation.
We're going to dive deep into CFTR in a moment because it ends up being the key to actually fixing this disease.
It really does.
But just to make sure I'm wrapping my head around the scale of this paradigm shift, we aren't just looking at alcohol damage anymore.
No, not
We've literally grown these biological avatars from 37 patients, and they prove this is a deeply intrinsic, genetic, and cellular disease. So we're at about the 5-minute mark. Let's do a quick recap for everyone listening.
Sounds good.
Bullet 1. Organoids are living biological avatars of a patient's pancreas.
And bullet 2. The biobank challenges the alcohol stigma, proving this cohort is predominantly genetic and idiopathic.
Which leads me to a question for you, the listener. Imagine having a miniature living version of your own pancreas stored in a lab. How would that change the way you view your own healthcare?
It's a powerful thought.
It really is. So if we have these 37 avatars, what do they actually look like when we zoom in? Does an alcohol-damaged pancreas act differently than a genetic mutation pancreas at the cellular level?
To answer that, the researchers used a technique called multi-omic profiling. They didn't just look at the DNA.
Because DNA is just the static architectural blueprint of the cell, right?
Exactly. Instead, they looked at transcriptomics.
Okay, transcriptomics is like intercepting the daily work orders being sent out to the cellular construction crew. It tells us exactly which genes are currently turned on, being read and actively producing proteins, and which ones are shut down in real time.
And when you are tracking the work orders of thousands of genes simultaneously across 37 different organoids, you get a mountain of data.
So how do you even read that?
To make sense of it, they used a mathematical algorithm called principal component analysis, or a PCA.
Visualizing a PCA map is actually really intuitive. I want you, the listener, to picture a giant map of cities.
But instead of plotting these cities by traditional longitude and latitude, PCA plots these biological samples based purely on their genetic similarity.
So if 2 dots on this map are close together, it means their gene expression, their daily work orders, are nearly identical.
And if they're far apart, they're operating under completely different biological rules.
And the map they generated is striking.
It really is. They took the top 500 most variable genes across all the samples. On the map, the chronic pancreatitis organoids, represented by orange dots, are grouped together on a completely different biological continent.
A completely different continent than the normal pancreas organoids, the green dots, and the normal adjacent tissue, the blue dots.
Right. In fact, principal component 1, which is the main horizontal axis of the map, accounts for a massive 38.87% of the total variance.
Meaning the single biggest difference between any 2 cells in this entire massive study Wasn't their age or their gender or their background.
No, it was simply whether they were diseased or healthy. The state of disease overwrites everything else.
But here's where it gets really fascinating. What happens when you zoom in on that disease continent of orange dots?
What's fascinating here is, through this multi-omic profiling, they discovered 3 distinct molecular subtypes of chronic pancreatitis.
The diseased avatars clustered into 3 specific groups.
Yes. But here is the absolute mind-bender. These 3 molecular subtypes are completely independent of etiology.
Wait, I want to make sure the gravity of that sinks in.
Oh.
You're saying the original trigger of the disease doesn't dictate the subtype.
Precisely. An avatar from an alcoholic patient and an avatar from a patient with a genetic PRSS1 mutation could land in the exact same subtype.
Wow, they could have the exact same molecular disease profile.
Exactly. The external trigger, whether it's years of heavy drinking or a broken gene you were born with, is just the spark that starts the fire.
But once the fire is burning, the cells adopt one of 3 highly specific, hard-coded inflammatory programs.
That's a great way to visualize it.
It's like saying it doesn't matter if a house fire was started by a lightning strike on the roof or a dropped candle in the living room.
Right. The way the wood burns, the way the drywall crumbles, and the way the house ultimately collapses follows the exact same physics and chemical reactions.
And the researchers didn't just find the fire. They pinpointed exactly where it's burning.
They used a volcano plot to do that. It's a graph used in statistics to show both the magnitude of a change and its statistical significance.
And what did the plot show?
The data clearly showed that the disease is highly specific to the ductal cells of the pancreas.
Right, because the pancreas is made of different specialized cell types. You have the acinar cells, which manufacture the digestive juices.
And you have the islet cells, which make hormones like insulin.
But the ductal cells form the plumbing. They are the intricate network of pipes that carry the digestive juice out of the pancreas and into the gut.
And the researchers proved that the plumbing itself is what becomes diseased. They didn't just guess, they looked closely at specific proteins marking these cells.
Like KRT19 and CA9, right?
Yes, and another crucial one called SOX9.
Think of SOX9 as a master control switch located deep inside the cell's nucleus.
Right, and when it gets flipped, it commands the cell to change its entire identity and behavior.
So in these diseased avatars, that SOX9 switch was jammed in the on position.
Completely jammed. It completely hijacked the duct cell's normal operating system.
And the transcriptomics backed this up, right?
Yeah, it showed that 732 different genes were significantly upregulated, meaning their volume was turned way up in these diseased ductal cells compared to healthy ones.
We're talking about genes like LINC025A2, which is a type of RNA that regulates other genes, and SLC10A2, which is a transporter.
Conversely, 238 genes were inappropriately turned down. The entire cellular program of the duct is corrupted.
Okay, we're at the 10-minute mark. Time for our mini recap.
Let's do it.
Bullet 1. Chronic pancreatitis completely rewires the ductal cell's gene expression compared to healthy tissue.
And bullet 2. There are 3 molecular subtypes of this disease, and the original cause doesn't dictate the subtype.
Which brings me to a big question for you, the listener. If the root cause of your disease doesn't dictate how your cells act once they're sick, does treating the cause even make sense anymore?
It's the multi-million-dollar question in medicine right now.
Because if the cells are running an independent, hard-coded disease program, regardless of how the fire started, shouldn't we be targeting the program itself?
Exactly. And that brings us directly to the intervention phase of this study.
So if we know that ductal cells are the ones misbehaving, and we know from the earlier genetic sequencing that a massive 27% of this specific cohort has a mutation in the CFTR gene, how do we fix it?
Well, let's clearly define what CFTR is first, because it's vital.
Right. Think of the CFTR protein like a microscopic dam or a molecular toll booth built right into the surface of these ductal cells.
Its primary job is to pump chloride ions, basically salt out of the cell and into the duct.
Now, a fundamental rule of biology is that water always chases salt through osmosis. So as the salt gets pumped out, water flushes through the pump right behind it.
And this water mixes with the digestive enzymes, keeping the pancreatic juice thin, watery, and flowing smoothly through the pipes.
But when that CFTR water pump is broken or missing because of the genetic mutation, the salt can't get out.
Therefore, the water doesn't follow.
So the pancreatic juice loses its liquid base and turns into a thick, sticky, inflammatory mucus.
It completely clogs the ducts. The digestive enzymes get trapped, they back up, they activate too early, and the pancreas begins to digest itself, triggering massive inflammation.
So If you are a researcher looking at these tiny microscopic organoid avatars in a dish, how do you actually test if their CFTR toll booths are working? You can't exactly look at them with a tiny wrench.
No, you can't. So they use a genuinely brilliant technique called the forskolin-induced swelling assay.
How does that work?
They take the organoids and expose them to a chemical compound called forskolin at a highly specific concentration of 20 micromolar.
Okay.
Forskolin acts like a chemical master key. It forcefully bypasses normal cellular signals and commands the cells to instantly open every single CFTR water pump they have to maximum capacity.
This visual is incredible. If the avatar is healthy and its pumps work, they all open at once.
Right. Salt rushes into the hollow center of the organoid, and water furiously chases the salt.
And under the microscope, you literally watch the organoid swell up like a water balloon filling from a hose.
It's amazing to see. But if the CFTR pump is broken, nothing happens. It just sits there, completely deflated.
It is a direct, undeniable physical proof of function. But this is where the data took a shocking turn, right?
Yeah, the researchers didn't just test the avatars that had known severe CFTR mutations. They also tested avatars that possessed what clinical geneticists have previously classified as non-pathogenic CFTR variants.
Wait, non-pathogenic means harmless? So according to current medical textbooks, those specific genetic variants are totally safe.
They shouldn't cause disease.
So if they're supposedly harmless, they should have passed the balloon test and swelled up, right? Did they?
No, they failed the balloon test entirely.
Here's where it gets really interesting.
Yeah, even though their DNA blueprint looked safe according to the current medical consensus, the functional swelling assay proved that their CFTR transport was severely impaired.
The avatars were practically deflated. They were diseased.
That is a staggering revelation. That means the medical community has likely been misdiagnosing people for decades.
We've been reading their genetic blueprint, seeing a variant we assumed was fine, and telling the patient they were clear, without ever physically checking if the actual biological house had running water.
Precisely. We relied on the map instead of looking at the territory.
Wow.
But the researchers didn't just watch these avatars fail and write a paper about it. They decided to intervene.
What did they do?
They took these diseased organoids and treated them with a class of drugs called CFTR modulators.
These are drugs already on the market, with names you might recognize if you follow respiratory medicine, right? Like ivacaftor, elexacaftor, tezacaftor, or lumacaftor.
Exactly.
How do they actually work on the pump?
They are mechanical fixers. These drugs physically bind to the defective CFTR protein structure.
So they, force it open?
Depending on the drug, yeah. They either forcefully prop the pump's door open so salt can flow, or they act like a molecular chaperone, helping a misshapen CFTR protein fold into the correct 3D shape so it can reach the cell surface in the first place.
And when they applied these modulators to the diseased pancreatitis avatars, did it work?
It worked beautifully. The swelling assay showed fully restored CFTR function. The water balloons inflated.
That's incredible.
But here's the most important part of the entire study. It wasn't just a mechanical plumbing fix. By restoring that chloride and water flow, it directly led to a massive decrease in inflammatory signaling deep inside the cell's nucleus.
We need to explain how profound that is. It wasn't just clearing a clog, it was turning off the disease program.
Exactly. Inside every cell, there are complex signaling pathways. Think of them like bucket brigades passing messages from the outer surface of the cell all the way down to the DNA in the nucleus.
And they measured this.
They used highly specific antibodies to measure the activity of these bucket brigades. They found that fixing the CFTR pump profoundly downregulated mitogenic signaling pathways.
We're talking about pathways with names like EGFR, STAT3, ACT, and ERK12.
Right. And when those specific pathways are overactive, they drive rapid chaotic cell division and massive stress responses.
But even more crucially, the researchers saw a dramatic decrease in a protein called phospho-STAT3.
Yes.
Let's visualize phospho-STAT3. Think of it as the cell's internal fire alarm system.
Good analogy. In a chronic inflammatory disease, that alarm is jammed on, constantly blaring, telling the cell it's under attack and needs to mount a defense.
Which ironically just causes more tissue damage.
Exactly. And the data proved that simply by fixing the water pump on the surface of the cell, they literally turned off the phospho-STAT3 fire alarm inside the nucleus.
Okay, 15-minute mark. Let's do our final mini recap.
Bullet 1. CFTR acts as a cellular water pump. When it fails, the pancreas inflames.
Bullet 2. The forskolin swelling assay physically proves if the pump works.
And bullet 3. Existing CFTR drugs can restore the pump and stop the inflammatory disease programs at a cellular level.
So I have to ask you, the listener, could a drug originally...
The data strongly suggests that for a highly specific, molecularly defined subset of patients, the answer is yes.
And we only know this because we finally had the living avatars to test the theory on.
Right.
So what does this all mean? When you step back and pull all of these incredibly dense scientific threads together, the 3D biobank, the transcriptomic map of cities, the swelling water balloons, the silenced fire alarms, what is the grand takeaway for the future of medicine?
If we synthesize this entire journey, this paper definitively proves that chronic pancreatitis is not just a passive organ reacting to external damage, like alcohol abuse.
Right. It's so much more than that.
These CP organoids, grown in a completely clean dish, totally isolated from the patient's body and any toxins, retained epithelial intrinsic inflammatory disease programs.
So the disease isn't happening to them anymore. The disease is fundamentally baked into the cells themselves.
They are intrinsically dysfunctional, but crucially, as the modulators proved, that intrinsic dysfunction is targetable.
We started this deep dive talking about a 35-year-old patient trapped in the murky, frustrating world of medical diagnoses, where the X-ray machine is useless, the assumptions are stigmatizing, and doctors are forced to guess.
And we just saw researchers use patient-derived avatars to successfully identify a molecular failure, test highly specific modulators, and literally fix diseased human pancreas cells in a petri dish.
It's unbelievable. So I want to leave you, the listener, with a final lingering question to ponder.
What's the question?
If we now have the technology to grow a living, breathing replica of your specific diseased organ, and we can use that avatar to safely test targeted drugs outside of your body, what is stopping us from doing this for every single chronic inflammatory disease before we ever prescribe a single pill to the actual patient?
The era of trial and error prescribing is coming to an end. The age of the biological avatar has officially begun.